First name is Kendall.
Okay, let's try that again. Good afternoon. I'm Dr. Kay Stefanik, Assistant Director of the Iowa Nutrient Research Center. I want to welcome you all to our spring seminar series, Insights from Next Generation Water Researchers. Each seminar this semester will have two speakers who are either current or recent graduate students or postdocs who have worked on INRC funded research projects. Now, before we begin, I have a few housekeeping items. First, this is a hybrid meeting. For those of you online, you can type your questions into the chat box, and I will ask them at the end of the presentations. For those of you who are in Ealing's Hall, we will have a time at the end of the talk where you can ask your questions directly. And then second, this will be recorded and posted to the INRC website.
Nope.
Now, for our first speaker, we have Philip Rockson. Philip is a PhD student in the Department of Agricultural and Biosystems Engineering at Iowa State University, working with Dr. Raj Raman and Dr. Dan Anderson. He will be presenting on cover cropping and application strategies for water quality and high yields.
Hi, good afternoon, everyone. So my presentation will be on cover cropping strategies combined with money application in order to maintain crop yields as well as increase the water quality. So first of all, I'd like to thank my advisors, Dr. Raj Raman and Dan Anderson, for helping me throughout this whole process. Also, I would like to thank the NEF research firm up in North Shore for helping us with the sample collection, as well as the Iowa Water Quality Research Lab with the analysis of our samples. I also did have my former colleague, Gabby, who started this project and handed it over to me. And I do have some undergrads helping out with the analysis. So this research is being funded, as she mentioned, by the INRC, also the Region PGC and Iowa POC Research. So a little bit about myself. I'm from Accra, Ghana in West Africa. I did my undergrad in KNUST, which is Kumasi, another city in Ghana. And after working for some time in the banking sector, I still felt the need for agriculture to be improved in Ghana. So it made me think, and when the opportunity opened up to pursue a master's here in Iowa, I picked it up. So thanks to Raj and Dan. So I moved here in the fall of 2022, did my master's, graduated last year, and started my PhD. So to start with, Iowa is the largest pork producer in the country. And one of the products or the waste product that comes out is the manure. We have done a good job trying to contain it and avoid any polluting the environment, but we can't store them indefinitely. It would definitely have to go somewhere at the end of the day. So this leads to my next slide, which are the counties here in Iowa and the ratio or the proportion of manure going into the fields. So you could see here in the west side where these counties barely have any manure going back into the field. But we see an increment, and in 2022, this is the current county map where the upper states have almost all their manure being produced going back into their farms. And these ones have almost zero manure going into their fields. So why the study? Iowa has a nutrients. Well, nitrogen by 41% and phosphorus by 29%. I think by the year 2035, we could probably maybe decide not to intensively farm, but the livelihoods of most Iowans depend on crop yields. We do need the soybean and the corn yields. So in order to address both stakeholders, farmers and the environmentalists who are concerned with the nitrates being leached in the environment, we would need multiple strategies to address this. So hence this study. So the goal of our research is to evaluate 2 perennial cover crops, which are Kentucky bluegrass and Redox hybrid babosa, as well as an annual crop, which is quite common here in Iowa, cereal rye, on its impact on nitrogen leaching. We believe those biological processes could complement each other and help reduce that. We also want to compare the application of manure during certain times, or the timing of those manure application treatments on the nitrates being leached from the environment. So a little overview of our project. It's located in Nashua, northeast of Iowa, and it's 36 plots with each almost an acre. And they do have, okay, I'll get there. So the experimental design was to set up a random complete block design, and each of them has been replicated 3 times. So for each of the plots, we have tiles underneath, 3 specifically. But in order not to have the results from the edge ones influencing the treatment of the other ones, we did sample just the middle tiles. So for our cover crop, as I mentioned earlier, we do have 2 perennial cover crops, the Kentucky bluegrass and the Redox hybrid babosa. For our annual cover crops, we did have 2 separate applications of it. We planted one early in the season and one late fall. I included pictures just to help. It was difficult for me to identify it, but I'm guessing you'll be able. So this is the cereal rye. This is the
Yeah.
It was not on his calendar.
Yeah, I don't, okay, yeah,
Just an FYI.
yeah, yeah,
But you know.
yeah.
Because I was
For the
looking for
manure
it,
application,
before spring break.
had 3
Yeah, why doesn't he?
treatment, the 4 applied manure, which had 100 pounds per acre. We also have the spring, which was 150 pounds per acre. And for our split application, we did 100 in the fall and 50 of UNI during the spring. So this leads to 12 treatments in total, and this is the complete list of our treatments. So we have the split application for the corn soybean rotation. For our manure in the spring, we also have the combination of corn and soybean. For the continuous corn, we did just the 4 manure application treatments. And all the plots were, okay, some of the plots were in no-till, which are the corn soybean rotation. However, the 4 manure application with continuous corn did have strip till. I did include some images here as well. So this is the Kentucky bluegrass treatment. And here's the early seeded rye. We did the seeding in September of last year. Here's the corn without the cereal rye, which will be done after harvesting in the fall. And here we have the RHB treatment. One thing I would like to point out is the Kentucky bluegrass. We did suppress it in 2020, and that hasn't fully recovered, and hence has some impact on the results we did have. So I'll move straight to the results for the 2024 season. We did have a cumulative precipitation last year, which was quite good for farmers, of almost 35 inches. And for our tiles, we had 33 samples. We could see that from August, we no longer see any tile flow. It's almost flatline. And this is a result of the corn either taking up most of the water or the water not getting deep down. So for our nitrates exports, in the spring, as expected, we saw higher nitrates leaching out of the soil or out of the plots compared to the summer, except for the Kentucky bluegrass treatment, and we suspect or we believe that because it had some cover, it was generally lower. But as it began to go dormant during the summer, the nitrate leaching kind of picked up. And these are just the raw values from the lab samples. So when we flow with the results with the total accumulation of the water coming out, we did see the total nitrates leaching out of the soil for the control treatment, which had just or no cover crop, was fairly higher than the RHB treatment in this case. And when we compare the reduction, it wasn't statistically significant. Although there's a numerical reduction, we do not see any statistically significant reduction. We believe it's as a result of the current state of the Kentucky bluegrass. We could barely see them. There are just some patches in the field compared to what we expect or what would be ideal. Hence, we plan to reestablish them during the spring. For the corn rotation, we see a fairly high output from the split treatment compared to the others as well, except in the cereal rye treatment where we see some reduction during the spring and more in the summer. Well, we think that, or we believe that probably maybe the cereal rye still had absorbed most of the nitrate from the plots before coming out from the spring season. We do see some significant reduction with our treatment here, which is the Redox hybrid Barbosa compared to the other three treatments, and it had the lowest nitrates coming out from the plots. When we also flow with this output as well, we see that there's statistically significant from the other treatments. The RHB had a reduction of 10 to 29% in total of the nitrates leaching out. So we've been running this for some year. We noticed that this was fairly higher than what we had in the previous year, which was 2023 and 2022. So we decided to kind of explore something that may be happening. So we think that during the 2023 season, which had very low precipitation, the nitrates in the soil profile moved when we received the rain, but couldn't get out of the tiles at the end of the day. When we compare that to the 2024 season, which had way more precipitation than the 2023 season, those nitrates was able to move out of the soil and came out of it, hence explaining why we saw the higher nitrates compared to the previous season. So this explains, because we, in the soybean treatment where we don't have any manure applied, we still see fairly higher than normal nitrates coming out. And we believe that accounts for it. So when in the soybean treatment, which had the cereal rye applied right after harvesting the corn before the previous season, we noticed that it's way below the recommended rate of 10 milligram per liter in the treatment that had cover crop. The RHB, due to the planting in the spring in March, couldn't do much in the reduction, although we do see some numerical reduction compared to the treatments that do not have any form of cover. So the spring application of the, the spring planting of the RHB didn't reduce it as much as we would want, but the treatment that had cereal rye during the fall has significant reduction. So we now want to compare the output to the yields from each of the plots. In our continuous corn plot for the Kentucky bluegrass application, or sorry, the treatment, the yields are almost comparable to the ones without any cover, which is our control treatment. The yields are almost similar. With the early seeded cereal rye, we also see the yields are almost equivalent to the one, or even higher than when we had no cover crop. For our RHB treatment, we did have some significant reduction in nitrates. We saw a yield loss, which was fairly lower than the other treatments. And when we analyzed it, was statistically significant, which means there would be, or there was a yield hit with our current treatment. For our soybean, which had also had the RHB, there was no reduction in yields. The ones that had cereal rye as well had almost comparable yields to each other. So to conclude, we do see nitrate reduction in treatment that have cover crop, such as the corn in the RHB or Redox hybrid Barbosa, and soybean and cereal rye. We studied that having continuous cover crop on our fields do impact the nitrates from the field. The yield loss we see in the RHB treatment kind of emphasizes why we do need to have a conforming PGC instance. In our treatment, we had a cover crop spread out the entire field without creating any cropping zones. Hence, we suspect that will be the reason why there was a yield hit. So in this year's treatment, you try to create a cropping zone in order to fit a corn in as well. For the manure application, the spring manure application had the lowest nitrates leaching out of the soil. And we believe that would be the ideal period to apply your manure. When you combine that with a cover crop, the effect is enhanced. And when we take some insights from the KBJ plots, which had like signal controlled ground cover, we did not see any yield hits. Hence, we believe that a well-controlled PGC system could maintain the yields we have. Thank you.
We do have time for maybe 2 questions.
You mentioned you're going to make your radix hybrid verbosa system conforming. Could you talk to a little bit more what that means and why someone made a terrible cropping decision this year?
Okay, so the best management practices agreed is to have a cropping zone separated from your grass strip. In our case, we spread out the RHB on the entire plot. I think I could probably just show a picture of it. Yeah, so it might not be too clear, but when you look at this picture, you see the grass on the entire field, which is not, you should have something similar to this when you have it. So that makes it not conforming because insurance wouldn't cover it. And you do get some benefits, but in order to have the most out of the field in terms of yield, you do need to make sure you have a cropping zone.
So you just mentioned insurance. So if the grass strip is a certain distance from the corn, then that means it would still be eligible for insurance.
Yeah, but currently the two, we're still in talks. Currently there are 2 cropping systems we're using, the Redux hybrid verbosa and Kentucky bluegrass still isn't covered, but cereal rye is covered under insurance currently.
Well, thank you. We're going to switch to our second presenter, but if you have any additional questions for Philip, please hold those to the end. Yeah, I'm trying to figure out how to switch the share screen. It's popping up on the screen.
Okay.
Okay, well, while we get technology sorted out, our second presenter is Taylor Ruman. Taylor is a PhD student in Dr. Michelle Sapir's lab in the Department of Agricultural and Biosystems Engineering at Iowa State University. She will be presenting on leveraging microbial communities to enhance woodchip and corncob bioreactor design and performance.
Hi everyone, I'm Taylor Broman. As she said, I'm a 3rd-year PhD student working with Dr. Michelle Sapir. All right, and today's work is also supported by master's student Lillian Chang and Dr. Adina Howe. Yeah, and I'm going to be talking about the microbial communities within woodchip and corncob bioreactors today.
If I get it wrong.
If we get moving.
There we go. Yep. Okay.
Beautiful.
Presenter mode.
Yeah. Now is it okay?
Oh, there we go. Okay. Perfect. All right. So as most of us in this room know, excess nutrients in Iowa waterways is a pretty big issue. I don't know how to get that. All right, I'm just gonna leave it. If we look at a map of the US here, in green, we're showing cropland. We can see that Iowa clearly is predominantly covered by green cropland. But at the same time, if we look at a map of soil drainage ratings, we also fall into that poorly drained, very poorly drained area. So in the 1800s, tile drainage was developed. It's that perforated tiling that lies about 3 feet or so below the soil surface and drains excess water from on top of the cornfields. So instead of letting that excess water sit there, it drains down through the tiles into a nearby waterway. But as, again, most of us know, the issue with this is that as we're draining the water, we also drain the excess nutrients as well, which likes to deliver a lot of excess nitrate down into the Gulf, creating an area that we often reference as the dead zone, which is an area of hypoxia. So we talk a lot about edge of field practices, and the one I'm going to be talking about today are woodchip or corncob bioreactors. So if we're looking at a diagram of an ag field, we can see here, this is our typical tile drainage, and instead of it dumping straight into a nearby waterway, we instead route it through an underground trench that's full of wood chips. For reference, about the size of a school bus. So instead of that water dumping right in, we allow the bacteria within the wood chips to act as an enhanced carbon source to sort of eat away at the nitrate that's available. Because since it's underground, there's not oxygen there to metabolize, it uses that nitrate instead. And if you haven't seen a woodchip bioreactor in the field before, this upper right-hand picture shows one under construction. I like to show it for a size reference, and that will continue to be filled up with some more wood chips and then topped off with the soil cap. So the goals and objectives of my project specifically are to determine key differences in the microbial communities between corncob and woodchip bioreactor environments that impact performance, with the objectives of identifying these key components of the microbial communities in order to optimize the design and performance of these woodchip and corncob bioreactors. So for my project here, we're using a controlled lab approach. We're not out in the field, and the setup is your stereotypical upflow columns. We have 6 across. These 3 are full of corn cobs, as you can see down the right-hand corner, and the ones on the right are full with wood chips. So the way that this works is that this gray influent tub is mixed up and filled with a nutrient solution that's mostly DI water, and then also diluted with a nutrient solution that we like to set at 30 milligrams per liter of nitrate in order to mimic tile drainage water. So that's what's being pumped and controlled at a set flow rate by this pump here, up through the columns, out through the top, into those white effluent buckets. And we chose to run these at 3 different flow rates, often called HRTs. So something to keep in mind, HRT is hydraulic residence time, so how long the water is actually in the system. So the lower the number, for example, the 4-hour that we ran it at, is actually moving a lot faster than our 16-hour, because at the 16-hour, the water's in there for 16 hours. Previous research has shown that 8 hours is your ideal condition for denitrification, but in the field, you can't really control how much rain you get and how much water is always going to your system. So we chose to run these at the 4-hour and 16 to account for dry and wet periods as well. And we ran these according to pore volumes. So we have 126 pore volumes of data for each aspect that we chose. For reference, that's about 3 weeks at a 4-hour HRT. So in order to break down how these columns perform, we broke it down into 3 categories: water quality, greenhouse gas production, and the microbiome DNA. So I'm going to start with everybody's favorite here, water quality. So in order to measure this, we collected the water samples at the end of the effluent. So not from within the system, but as it's trickling out into the bucket, We collected those samples and analyzed them in the lab. And we looked at nitrate, nitrite, total organic carbon, and ammonia. But today I'm just going to be talking about nitrate. So looking at the setup of this figure here, as you're looking at the columns, 6 columns across, that's how this data is presented. Just from left to right, you can see we don't have data for 4-hour HRT for corncobs yet. That's going to be run later this month. But we have 4-hour for the wood chips here, and then 8-hour and 16 going across, and it's shown in nitrate percent removal. Since we know exactly what content of nitrate is going in, we can calculate what goes out and then get the percent removal from there. The first thing that we can see is that the corncobs removed a lot more nitrate than the wood chips at both the 8 and 16-hour HRT. And you might notice that this column right here wasn't really performing well at the 8-hour, but then at the 16, it jumped right up. And that's just because at the end of the 3 months of running the 8-hour HRT, the system was so clogged that we couldn't actually get water through it. So I had to dump all of them, replace with fresh corn cobs. And after we replaced them with new ones, it seemed to perform just like the rest. So not really sure what was going on there. Hopefully we'll have an answer by the time I'm done with this project. But then you can also see that the wood chips didn't really vary in performance too much across the HRTs, kind of like we had hoped. So the short answer to how does nitrate removal vary between carbon sources and across flow rates is that corncobs remove nitrate better at the 8 and 16-hour HRTs so far than the wood chips. And both the wood chips and corncobs seem to remove nitrate a bit better at 16-hour HRT than 8, although the difference really isn't that much. And then moving on to our second parameter here, dissolved greenhouse gas sample collection. So the first thing I want to point out is that this is a dissolved gas sample. It's not a straight air sample. You're actually getting the gas value from a water sample. So excuse the blurry picture here. You probably can't tell, but there is a little stop valve right here attached to the cap. And then this big black piece is an air vent. We had to put vents on there because at first the tops of these columns were popping off even after being drilled on because of the amount of gas being produced. But adding a vent in here raised the water level to up here. So our interaction with the air from the water is above. We were actually collecting the sample, which is down beneath the cap. And all of this is done assuming that once the water is discharged from a tile or from the effluent tube in our lab case, that all dissolved gases will release into the atmosphere. And just as to why we're collecting this data, our goal is to improve water quality with bioreactors, and we want to avoid producing any other pollutants, especially at flow rates that are too quick or way too slow. So for dissolved gases, we looked at carbon dioxide, nitrous oxide, and methane. And our data here is set up pretty similarly, except we have the averages of each 1 through 3 and 4 through 6 for the corn cobs and wood chips. Again, we only have the 1 wood chips for 4-hour. And the first thing I want to point out is our scale, because at first glance, that wood chip one looks like it's way higher than the rest, but our scale only goes up to 1.5 grams of our carbon dioxide equivalent per cubic meter per day. So that's a very small number. And this is actually a stacked graph, believe it or not. I didn't just leave out the other gases. But the carbon dioxide and nitrous oxide levels are so small that it's pretty much negligible across all flow rates so far and carbon sources. But I do think that this is interesting because we would expect higher nitrous oxide rates at a 4-hour HRT because that would lead to incomplete denitrification, but we really aren't seeing that so far in our results. And then next, kind of the novelty piece of this project is looking at the microbial DNA. So to talk about that today, I'm going to be talking about the bacterial sampling. And just a couple notes before diving into the data here. The woodchip and corncob DNA samples are taken from ground samples. So they're not taken from these full wood chips as shown on here. I actually put some corn cobs and wood chips into a coffee grinder, ground them up, put them in little mesh party bags, put them on top of the columns before putting the cap on. So they're saturated and treated like all the other material in the system, but this way we had a greater surface area exposed to the water. So this just helped with all aspects of DNA extraction. And another key point is that we're looking at the bacteria attached to the wood chip and corn cob. We're not actually looking at DNA from the corn cob or wood chip. And why we're looking at the microbiome, it's key to unlocking bioreactor efficiency. There's a lot of work done out there with different flow rates, temperatures, and physical conditions. But when we get to actually understanding who is doing the work and when and why, that's when we can really improve system efficiency. And just a quick note that when I'm saying bacterial sequencing as to what that actually is, I just want you to think of it as a term of a survey of who is in the sample and how many of them are actually in there. So the workflow for this aspect is we're collecting the ground sample from the top. I guess I didn't say this, but for the water sample, this and gas data, that was weekly at 8 and 16-hour HRT, about every other day for the 4-hour. So we have our ground material, we store it in the freezer, we do our DNA extraction, and then we get to the sequencing part. So I want everybody here to kind of picture, it's Halloween. Everybody wants some sweets. It's candy time, okay? You have two people come up to you, they have huge bags full of candy, and you want to know what's in them, but you don't feel like digging through and looking at every single piece. You basically just want a survey of what's in there. So that's kind of what we're looking at here. So before just getting overwhelmed with the data, I want you to picture that corn cobs is one bag, wood chips is another bag. We can see that just looking at the colors, there's a really big difference in what candy is in each bag, right? So we can see that the corn cobs is made up of a lot of different pieces than the wood chips. Those orange and red colors compared to the green, and both are 8-hour and 16-hour. We can see with the 16-hour, there's a couple of similarities with that gray color there, but the relative abundance of what's actually in the sample has a pretty big difference visually across both of them. And this is just pretty much showing what is different in the sample versus our next figure is going to show us how they're different. So again, going back to the trick-or-treating example, if you were to look at these bags, one of them, it looks like one kid went to a place where they got a lot of small pieces of candy. They got all the miniature and fun size. Whereas the other person, it seems that their survey shows that they got a lot of the big size, the really big candy bars. And you would have assumed that this would show some predictable pattern of where they actually went trick-or-treating, which is the same as what's happening in our corn cob and wood chip samples. And then next, I'm going to show you a graph called the principal coordinates analysis, essentially just showing us how different they really are from each other on a quantitative scale instead of just looking at some colors going on. So this axis here, we have 8-hour and 16 again. The orange color is your corn cob, whereas the green is your wood chip. So we're looking at variability within the samples. So we can see that our corn cob samples are way over here and our wood chips are over there. So that's just showing us that there's a really big difference, and that one kid got a lot of mini, one kid got a lot of large, and they're really different from each other. And then looking at the 16-hour, we can see that the corn cobs have a wide variability within themselves. There's a really wide range of samples. So again, this could look like the one corn cob person, the one trick-or-treating bag had a lot of different types of candy, although they're all small, versus the wood chips are really tight-knit, so they got a lot of the same candy bar over and over. So our key points with this are that the bacterial communities in corn cobs have more variation and a higher diversity than within the wood chips, which could point to more resilience to outside factors. And bacterial communities are significantly different between the corn cob and wood chips and between the 8 and 16-hour. So to answer the question, how do these microbial communities differ between the carbon sources and across all flow rates? We'll be adding 4-hour on there soon enough. There is a significant difference across all flow rates and carbon sources, which is really exciting for us to see. And so kind of just to do an overall analysis here of the overall performance, wood chips versus corn cobs. We did see so far that corn cobs are removing nitrate better. And that carbon dioxide and nitrous oxide values for both, even though they're both so low that they're both get the green check mark. And even though corn cobs didn't produce as much methane as the wood chips, the value for wood chips was still really, really low. And then for the microbiome, this is a piece that we're still really digging into here to see exactly what the difference is, why is this different, and how does it relate to performance. But a side note for now is that the corn cobs do have more diversity in what we've seen, and that again, this higher diversity can lead to more resilience in field situations. So with that, I will take any questions.
You said that corn cobs in the 8-hour HRT on that one column was all plugged.
Yeah.
Is that breakdown of the corn cobs or film or what?
I believe it was film. I have some pictures somewhere of when we dumped them that they were pretty much like all stuck together versus the first two columns when I dumped them, they just spread apart, which we weren't originally looking to do any sort of fungal analysis on this data. But we went ahead and added in the ITS gene for qPCR, which is just the fungal marker, and then also looking at the AMF fungal marker data for the sequencing as well, because we were really curious about that.
How does that tendency to plug affect how you compare those two packings? I mean, it looks like corn cobs win, but how frequently are you going to have to deal with the problems so caused?
Right, and I think one thing to note that with this, our samples are saturated 100% all the time, and they're broken down into smaller pieces already just to fit in the column. So for that, I don't know if it would be a good example for field situations. But yeah, for our 16-hour, I thought we were going to have problems running through a full 3 months because I thought surely if they clogged to 8 hours, slower flow rate would lead to more fungal growth. But we didn't have to. So I'm curious with our 4-hour if that's going to be a problem.
We have a couple questions online. For the corn cobs, are those straight corn cobs or are they mixed with leaves and stalks?
Straight corn cobs, those from the Biocentury research farm.
What is the difference between wood chips and corn cobs in terms of lifespan?
As far as lasting in the reactor, I don't think we really have a set answer to that yet. We do have at our pilot scale bioreactors, we have some that are 75%, 25% corn cob wood chips, both ratios. And they've been out there for 6 years and are still performing better than the 100% wood chip reactors that are out there. So far, they've been lasting longer than I think we expected. But there aren't really 100% corn cob reactors out there yet to really have a good answer to that question.
Interesting project you have here. Thanks for your presentation. I'm curious about the gas data. What do you think is blowing the top off of those?
Yeah.
And I understand that it's you're putting the CO2 equivalents, but I'm curious as to why we can't see CO2 at all in this plot. How do we get such a high ratio of that thing CO2?
Yeah. So for the first question, the blowing the tops off, that only happened with the corn cobs. And I think it could have been just because there's so much carbon and it's in such a small confined space and again, completely saturated that there is a lot. Actually, I think I have a chart hidden back here somewhere. Oh, I didn't mean to do that. And I think it probably would relate to our total organic carbon. which I haven't completed the analysis on it yet. But we can see an initial flush of total carbon at the beginning, especially in the corncob. So I'm guessing there's something to do there with just the breakdown and the initial decomposition of that. And then as far as your other question, I think that's a great one and something that we're also very interested in.
So is it only the, so you're only looking at what's dissolved?
Correct.
And you're venting some off as well?
The vent is above where we're collecting the sample from. So the idea is that that vent up top doesn't play any role in the actual sample collecting. If we were to collect right at the surface, yeah, probably.
Okay, but the whole system is vented. Yeah.
Correct. Yes.
All right, that's what I.
And again, important. We collected the gas really to correspond it with the microbial communities that are actually doing the work. And again, completely saturated system. I don't know if this is like, that's the gas data, if it scales up correctly to field reactors. Yeah.
Thank you.
Yeah.
I think the corn cobs are serving as better candies for bacteria.
That's what it seems like.
If you look at some sugar contents in the corn cob,
True.
that might answer both the
Why
questions.
don't, yeah. Yes, Dan.
You pointed out that here you ran nice steady state to cause microbial differences. And you alluded to the fact that in your field bioreactors, you actually get fluctuating flow rates. When you talk about resilience, I wonder, are they more resilient to those fluctuating flow rates? And how do we get at that aspect of the question?
That's a really good point. I know there has been some work done. We were looking today, like doing flushing of systems, I think, and then looking at the gases that way. Yeah, it was interesting that in the, oh, I think I skipped past the one I want. That in the slower flow rate, that I guess the corn cobs, the range of species in there was greater at both flow rates, but at that 16-hour, it was greater. Sounds like another good project to do, is what I think.
And then we have another question online. Did you have to inoculate the columns to ensure there were denitrifiers present?
Yeah, so what we did before all of the 8 and 16 and 4-hour, we went and collected tile water, and we let that sit just in there, saturated, not moving for a week, just to make sure that they had the microbial communities that were present. And we tested the nitrate over that week to make sure that the removal was actually happening. And that seemed to work pretty well.
But just inoculated with regular old tile.
Yep. Yeah, we did some little batch tests of using corn cobs and wood chips from the field with our nutrient solution. But the tile water was both easier and worked better. So.
But the water you pumped through there was not tile water.
Correct. The only time we used the tile water was sitting in there for the week, and then we flushed it through with the influent and nutrient solution that's made in the lab.
In the color map where you showed the different microbial communities, so it looks like there's structurally the communities are different. Are they also different functionally, or are they using similar compounds?
I think that's our next step of what we're going for. We are, at least I'm planning to do, I've done some qPCR for specific denitrifiers and specific nitrous oxide emitting genes. And we don't know if any of that is, if we're picking the right piece that we're looking at for what genes we're targeting. But yeah, that is kind of the next step that I'm excited, and what Lillian's also working on.
All right, we still have about 5 minutes, so maybe at this point we could open up the floor for questions for both Taylor as well as Philip, if there are any additional questions for Philip. All right, well, we don't have any more online. If there aren't any more in the room, we'll end a little bit early then. I do have a quick announcement for our next seminar. It's going to be April 9th. We have Kelsey Farron talking about this campus under future climate conditions. And then Shushan Du talking about dynamics of conservation practice continuity in Iowa agriculture. April 9th, same room or online. Okay, once again, I want to thank our presenters.